This is the BRCA Lab in AP Bio by Emma Karakaran and Anthony Alito. We are going to be exploring interdisciplinary connections between the BRCA gene and AP biology unit connections. What was the lab? The purpose was to figure out if Hannah had breast cancer. The method was through gel electrophoresis of Hannah and her family's DNA to determine the mutated allele, BRCA, causing the cancer. The result was that because of her mom and aunt were confirmed to have the cancer, we can now determine that Hannah also has it because they share the same allele.
So, Unit 1 mostly talked about the four biomolecules, the macromolecules, which are carbohydrates, lipids, proteins, and nucleic acids. And they're made up of life elements C, H, O, N, P, and S, and in some kind of combination. And one of those we can focus on is DNA. And they are made of nucleotide monomers, so DNA is the polymer. And each nucleotide monomer has a certain hydrogen base, which can be adenine, thymine, guanine, or cytosine. And each nucleotide monomer, depending on which base they have, creates a nucleotide sequence, and that's how DNA stores its info. Another macromolecule we can look at is protein, which is a polymer made of amino acid monomers. And the folding of that amino acid monomer chain, called a polypeptide chain, that is the structure which dictates its function. So, for example, in enzymes, it has a very specific structure that allows it to do its certain function. In our lab, we used restriction enzymes to cut DNA segments. So we need to cut the DNA segments of the family to be able to run it through the gel electrophoresis. And the shape of that enzyme, dictated by its the way it's folded, allows that active site to have a certain shape where it will only bind to a certain nucleotide sequence in the DNA to cut it at that place. And that allows it to cut it at the exact place we want so that the alleles will separate into different segments.
Unit 2 was about cell structure and function. Cells have many parts and different jobs. Kinase function in cancer cells helps develop resistance. The use of plasma membrane proteins regulates cell metabolism while facilitating information between cells. It also plays part in oncogenesis via endocytosis and signaling. But by targeting certain membrane proteins, it can stop cancer growth. Various cancers share use of similar membrane proteins, as seen in the photo to the right. Treatment plans then try to specify tumors involved. Unit 3 was about cellular energetics. Cells use the process of glycolysis to create energy. Altercations in signaling pathways can lead to tumors. But how do cancer cells grow and replicate? They use an increase of aerobic glycolysis, which is also called the Warburg effect. It's a mechanism that supports cancer cells' multiplication, as seen in the photo to the right. This results in a loss of tumor suppressors like BRCA1. Without suppressors, cancer cells can proliferate uncontrollably, meaning that they can grow and rapidly divide uncontrollably without the tumor suppressors being able to stop it. Unit 4 was about the cell cycle and cell communication. In this unit, we learned about cell growth in the cell cycle and signal transduction. BRCA genes send signals to begin creating suppressors. BRCA1 and 2 are tumor suppressor proteins that repair DNA. Tumor suppressors regulate the cell cycle, as shown to the right in the photo, to stop cancer. A result of this in the lab was biomarker testing. Biomarker testing is used to detect any mutations or causes of cancer. Biomarking for specifics is also better than DNA sequencing, as it is cheaper and way faster. Unit 5 was about heredity. In this unit, we learned about the passing on of traits from parent to offspring. In this lab, we learned about the possible inheritance of the BRCA1 and 2 and or 2 mutations from parents. We also learned about family history. In the lab, the Alterman family's history of breast cancer was noted, and this was used to create pedigrees and Punnett squares, as shown to the right. For pedigree models show family patterns of autosomal dominance for breast cancer, as seen in the top right photo. Multiple generations were included in the pedigree, as it went from the first generation all the way to the fourth generation. Punnett squares can predict the likelihood of inheriting breast cancer, as seen in the one photo to the bottom right.
So, Unit 6 is about gene regulation and expression, and also the concept of our body goes from DNA to RNA to protein, and that DNA is also replicated. Additionally, gene regulation happens in bacteria with operons and in eukaryotes with transcription factors, and we can find that in genetic engineering. One such method is gel electrophoresis, in which we create an electrical field that moves charged particles. Our charged particle is DNA, and because DNA has a phosphate group that's negative, it's overall negative, and it is going towards the positive electrode because negative and positive attract. And we create this electrical field with that buffer, which then runs charge through the electrodes, making one negative and one positive. So we load in the DNA at the negative, and it goes towards the positive. And because we cut these pieces of DNA into different fragments, the larger ones will take longer to reach compared to the shorter ones that will move faster. And that's how you can see which fragments are in the batch of DNA that you're running through the gel electrophoresis. In our lab, we use this to determine who had the BRCA allele, or so we know which allele is the BRCA allele and who had them. And we compare that to an allele ladder, which is kind of like a key of all the alleles that are there. Unit 7 explored the mechanisms of evolution with biological evidence in our structure, like homologous and analogous structures. Additionally, it talked about speciation from our ancestors, which we can draw out in a phylogenetic tree. One mechanism of evolution is genetic drift, which reduces genetic diversity, and it eliminates alleles until some of them become fixed and more constant. One type of genetic drift is founder effect, and it's caused when you have a few ancestors that are reproducing and creating a colony. There are 3 founder mutations in Ashkenazi Jews that are in the BRCA 1 and 2 alleles. And Ashkenazi Jews are Eastern European Jews who were more geographically isolated, and they also had endogamy, which means they married within themselves, and that's another cultural isolation factor, and that causes them to be more isolated and reduced genetic diversity. Additionally, we find that other mutations may also be present in Ashkenazi Jews that are causing breast cancer, such as the CHEK2 gene, which is also has some studies that show that it is another factor that can show and hint at breast cancer. Unit 8 talked about ecology, so different behaviors of species, like, for example, their own fixed pattern behaviors, as well as how they interact in a community, like predation, mutualism, parasitism, and things like that. Additionally, we see the diversity in ecosystems with many different types of species, as well as the transfer of energy, which can be demonstrated in a food web. Biomagnification is the process where pollutants they become more present in an organism as you go up through the trophic levels. So, like the consumers more and more get these pollutants in their body. One such example is estrogen bioaccumulation, and we saw there was a study in zebrafish that it was accumulating because estrogen is found in oral contraceptives and it's hard to remove from the water. So it's accumulating in the zebrafish, and so then it's accumulating in lots of marine organisms. Additionally, estrogen affects breast cancer because it fuels growth of like mitosis of the breast cancer cells, and so it fuels rapid growth with no stopping, as well as some studies have shown that some binding sites of estrogen are correlated with DNA breaks at those points, which are some new studies. So that's how estrogen, if it's bioaccumulating in organisms that we eat, it is being biomagnified in our bodies as well. So, some problems with bioethics is racism in the hospital because doctors are shown to have implicit bias, as everyone does. And some say that it is important to report racial backgrounds to have better diagnoses because, for example, genetic factors have been linked to race, such as the Ashkenazi Jew that we discussed. Additionally, knowing a patient's background can help understand them and personalize their experience to have a better recovery. Additionally, Learning a medical history makes more accurate decisions, as well as helps with further studies on race connected to diseases. Um, Also, some say reporting race furthers racism because they are treated differently with giving them less or wrong medications. Um, they have disease stereotyping with certain races, and they're unable to access insurance, even though some states, some law allows them to. Lots of states have not accepted that. Additionally, they have longer wait times for things like testing, like EKGs, less doctor availability in zip codes with higher minority groups, as well as higher disease mortality rates, even like infant mortality rates. It's proven that it's more common in minority groups.
Our second bioethics slide was on the concern about patients' comfort in diagnosis/treatment of breast cancer. In the first perspective, we see that it is important to be informed of cancer detection, as it gives patients space to digest the hard news and also offers time to discuss possible treatment options. It also helps being well prepared for devastating news as it brings confidence. Patients become well educated on breast cancer by learning about the cancer that they have. Insurance lets patients focus solely on defeating the diagnosis, and love and support from their close ones increases hope of defeating it. On the other hand, the second perspective is more of a negative side of the concern, as patients' levels of discomfort will rise highly with the diagnosis. Heavy anxiety and fear could arise. Depression due to heartbreaking news of cancer can happen, and patients' lives might change with the cancer existing. Patients' trust may also start to fall. Not having enough confidence in doctors or treatment could be one option, or they may begin to see financial issues due to the cost of treatment. Opinions and news on social media can also affect the patient mentally, and the lack of familiarity with cancer can leave the patient questioning.